The super-resolution discrete molecular imaging method is based on DNA paint technology, which uses so-called imager strands, dye-conjugated DNA probes, to detect complementary so-called docking strands that are part of the structure the scientists want to visualize. The docking strands are sticking out of a synthetic DNA origami nanostructure on a cover glass slip and are arranged in a triangular lattice pattern with 5 nanometer point-to-point distance. Because of their sequence complementarity, the imager strands bind transiently to individual docking strands within the DNA origami. The interaction brings the dye into the imaging range of the total internal reflection fluorescent microscope and produces a single molecule blinking event before the DNA probe detaches from its target. The diffusing DNA probes repeat their transient binding to different DNA targets in the origami. Movies collecting a series of single molecular blinking events are processed to identify their centers, which indicate the precise locations of the docking strands in the origami and their tight packaging in different patterns. The localization with super high precision of less than 1 nanometer, together with the microscope stage drift correction at less than 1 nanometer accuracy, enables the DMI-enhanced technology to resolve individual targets in a densely packed biomolecular cluster. The super-resolution discrete molecular imaging method is also able to detect docking strands with different sequences, which are indicated with different colors in the origami structure. First, imager strands complementary to one kind of docking strands are applied to image their targets. After washing the first type of imager strand away, a second and third type of imager strands are added one after another to image the other 2 docking strands sequentially. After processing the 3 imaging results, the spatial pattern of the 3 docking strands in the DNA origami is revealed at 5 nanometer resolution. The highly multiplex super-resolution imaging is achieved with only a single dye and a single optical path, which are artificially colored after the actual experiment is done.